Chapter 8, Cell, the Unit of Life. When you look around, you see both living and non living things. You must have wondered and asked yourself what it is that makes an organism living, or what it is that an inanimate thing does not have, which a living thing has. The answer to this is the presence of the basic unit of life, the cell, in all living organisms. All organisms are composed of cells. Some are composed of a single cell, are called unicellular organisms, while other like us, composed of many cells, are called multicellular organisms. 8.1, what is cell? Unicellular organisms are capable of first, independent existence, and second, performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is a fundamental structure and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to an electron microscope revealed all the structural details of the cell. 8.2, Cell Theory. In 1938, Matthias Claden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells, which form the tissues of plants. At about the same time, Theodore Schwann, 1839, a British zoologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is unique character of the plant cell. On the basis of this, Schwann proposed that hypothesis that the bodies of animals and plants are composed of cells and products of cell. Scladen and Schwann together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow in 1855 first explained the cell divided and new cells are formed from pre-existing cells. Omnis cellula e cellula. He modified the hypothesis of Scladen and Schwann to give the cell theory a final shape. Cell theory, as understood today, is all living organisms are composed of cells and product of cell. Second, all cells arise from pre-existing cells. 8.3, An overview of cell. You have earlier observed cell in an onion peel and or human cheek, cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane bound structure called nucleus. This nucleus contains the chromosome, which in turn contains the genetic material called DNA. Cells that have membrane bound nuclei are called eukaryotic, whereas cells that lacks a membrane bound nucleus are called prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plant and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Beside the nucleus, the eukaryotic cells have other membrane-bound distinct structures called organelles like endoplasmic reticulum, the Golgi complex, lysozymes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribozymes are found not only in the cytoplasm but also within the two organelles, chloroplast in plants and mitochondria and on rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelle called centrosome which help in cell division. Cells differ greatly in size, shape and activities. For example, mycoplasm, the smallest cells are 0.3 micrometer in length, while bacteria could be 3 to 4 micrometer. The largest isolated single cell is this egg of ostrich. Among multicellular organisms, human red blood cells are about 7.0 micro in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. 8.4 Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, pneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in size and shape. The four basic shapes of bacteria are bacillus, rod-like, coccus, spherical, vibrio, comma-shaped, and spirillum, spiral. The organization of the prokaryotic cells is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and function. All prokaryotes have a single wall surrounding the cell membrane except in mycoplasma. The fluid matrix filling the cell is the cytoplasm. There is no well defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to genomic DNA, the single centrosome, chromosome, circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistant to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in the eukaryotes are found in prokaryotic cells except for ribosomes. Prokaryotes have something unique in the form of inclusions. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. They are essentially infoldings of cell membrane. 8.4.1 Cell envelopes and modification. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure that is the outermost glycocalyx followed by the cell wall and then the plasma membrane. Although each layer of envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in the cell envelopes and the manner in which they respond to the staining procedure developed by Gram-Vis. Those that take up the Gram strain are called Gram-positive, and the others that do not take up the strain are called Gram-negative bacteria. Glycocalyx differs in composition and thickness among different bacteria. It could be a loose sheath called the slime layer in some, while in some others it might be thick and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extensions of plasma membrane into the cells. These extensions are in form of vesicles, tubules, and lamellae. They help in cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration secretion processes to increase the cell area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, these are other membranous extensions into the cytoplasm called chromatophores, which contains pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extensions from the cell wall called flagella. Bacteria show a range in number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Beside flagella, pili and fimbriae are also surface structure of the bacteria, but do not play any role in motility. The pili are elongated tubular structures made up of special proteins. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to the rocks in streams and also to the host tissues. 8.4.2 Ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cells. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, 50S and 30S units, which when present together form 70S prokaryotic ribozymes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysomes. The ribosomes of a polysome translate the mRNA to proteins. Inclusion bodies. Reserved material in the prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. Example: phosphate granules, cyanophysin granules, and glycogen granules. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. 8.5 Eukaryotic cells. The eukaryotic includes all the protists, plants, and animals and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane bound organelles. Eukaryotic, eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeleton structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possesses cell walls, plastids, and a large central vacuole, which are absent in animal cells. On the other hand, animal cells have centrioles, which are absent in almost all plant cells. Let us now look at individual cell organelles to understand the structure and function. 8.5.1 Cell membrane. The detailed structure of the cell membrane was studied only after the advent of the electron microscope in 1950s. Meanwhile, the chemical studies on the cell membrane, especially in human red blood cell, enabled the scientists to deduce the possible structure of plasma membrane. The study showed that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer sides and the hydrophobic tails towards the inner part. This ensures that the non-polar tail of saturated hydrocarbons is protected from the aqueous environment. In addition to the phospholipids, membrane also contains cholesterol. Later biochemical investigation clearly revealed that the cell membranes also possess protein and carbohydrate. The ratio of the protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocytes have approximately 52% protein and 40% lipids. Depending on the ease of extraction, membrane proteins can be classified as integral and peripheral. Peripheral proteins lie on the surface of the protein, while the integral proteins are partially or totally buried in the membrane. As improved model of the structure of cell membrane was proposed by Singer and Nicholson in 1972, widely accepted as fluid mosaic model. According to this, the quasi-fluid nature of the lipid enables lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of the functions like cell growth, formation of intercellular junction, secretion, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly or across the membrane without any requirement of energy, and this is called passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is, from the higher concentration to the lower. Water may also move across the membrane from the higher to the lower concentration. Movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require carrier proteins of the membrane to facilitate the transport across the membrane. A few ions or molecules are transported across the membrane against the concentration gradient, that is, from lower to higher concentration. Such a transport is an energy dependent process in which ATP is utilized and is called active transport. Example: sodium potassium pump. 8.5.2 Cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protects the cell from mechanical damage and infection, it also helps in cell-to-cell interaction and provides barrier to undesirable macromolecules. Algae have cell wall made up of cellulose, galactans, mannans, and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins, and proteins. The cell wall of a young plant cell, the primary wall, is capable of growth, which gradually diminishes as the cell matures and secondary wall is formed on the inner towards membrane side of the cell. The middle lamella is a layer mainly consisting of calcium pectate, which holds or glues the different neighboring cells together. The cell wall and middle lamellae may be traversed by plasmodesmata, which connect the cytoplasm of neighboring cells. 8.5.3 Endomembrane system. While each of the membranous organelles is distinct in terms of structure and function, many of these are considered together as endomembrane system because of their functions are coordinated. The endomembrane system include endoplasmic reticulum, Golgi complex, lysozymes, and vacuoles. Since the function of mitochondria, chloroplast, and peroxisomes are not coordinated with the above components, these are not considered as part of endomembrane system. 8.5.3.1 The endoplasmic reticulum. Electron microscope studies of eukaryotic cells reveal the presence of a network of reticulum of tiny tubular structures scattered in the cytoplasm that is called the endoplasmic reticulum. Hence, ER divides the intracellular spaces into two distinct compartments, that is luminal and external luminal compartments. The ER often shows ribosomes attached to the outer surface. The endoplasmic reticulum bearing ribosomes on the surface is called rough endoplasmic reticulum. In the absence of the ribosomes, they appear smooth and are called smooth endoplasmic reticulum. Rough endoplasmic reticulum is frequently observed in the cell actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for synthesis of lipid. In animal cells, lipid-like steroidal hormones are synthesized in SER, that is, smooth endoplasmic reticulum. 8.5.3.2 Golgi apparatus. Camillo Golgi in 1898 first observed densely stained reticular structure near the nucleus. These were later named Golgi bodies after him. They consist of many flat, disc-shaped sacs or cisternae of 0.4 micrometer to 1.0 micrometer diameter. These are stacked parallel to each other. Varied number of cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convexes or the forming phase and the concave trans or the maturing phase. The cis and the trans phases of the organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Materials to be packaged in the form of vesicles from the ER fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remains in the close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. Golgi apparatus is the most important site of formation of glycoproteins and glycolipids. 8.5.3.3 Lysosomes. These are the membrane-bound vesicular structure formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes, that is hydrolases like lipases, proteases, carbohydrases, optimally active at the acid pH. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. 8.5.3.4 Vacuoles. The vacuole is the membrane-bound space found in cytoplasm. It contains water, sap, excretory product, and other material not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In plant cell, the vacuole can occupy up to 90% of volume of the cell. In plants, the tonoplast facilitate the transport of a number of ions and other material against concentration gradient into the vacuole. Hence, the concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protist, food vacuoles are formed by engulfing the food particles. 8.5.4 Mitochondria. Mitochondria singular, mitochondrion. Unless specifically stained are not easily visible under the microscope, the number of mitochondria per cell is variably depending on the physiological activities of the cell. In terms of shape and size also, considerable degree of variability is observed. Typically, it is a sausage shape or cylindrically having a diameter of 0.1 to 1.0 micrometer, average 0.5 micrometer, and length 1.0 to 4.1 micrometer. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments. That is the outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of enfoldings called the cristae, singular crista, towards the matrix. The cristae increases the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the site of aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called powerhouses of the cell. The matrix also possesses single circular DNA molecule, a few RNA molecules, ribosomes, 70S, and components required for the synthesis of proteins. The mitochondria divide by fission. 8.5.5 Plastids. Plastids are found in all plant cells and in euglenites. They are easily observed under the microscopes and they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on the types of pigment, plastids can be classified into chloroplast, chromoplast, and leucoplast. The chloroplast contains chlorophyll and carotenoid pigments, which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigments are carotene, xanthophylls, and others are present. This gives the part of the plant a yellow, orange, or a red color. The leucoplasts are the colorless plastids of varied shapes and sizes, which store nutrients. Amyloplasts store carbohydrates, starch, etc. Potato. Example, potato. Elioplast stores oil and fats. The aleuroplast stores protein. Majority of the chloroplast of the green plants are found in mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles, like variable length 5 to 10 micrometers and width 2 to 4 micrometers. Their number varies from 1 per cell of Clydomonas, a green algae, to 20 to 40 cells in mesophills. Like mitochondria, the chloroplasts are also double membrane bound. Of the two of the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma. Thylakoids are arranged in stacks like piles of coins called grana, singular granum, or the integranal thylakoids. In addition, there are flat membranous tubules called the stroma lamellae connecting the thylakoids of the different grana. The membrane of the thylakoids encloses a space called the lumen. The stroma of the chloroplast contains enzyme required for the synthesis of carbohydrates and protein. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoids. The ribosomes of the chloroplast are small as 70S and cytoplasmic ribosomes 80S. 8.5.6 Ribosomes. Ribosomes are the granular structure first observed under the electron microscope as dense particles by George Pellet in 1953. They are composed of ribonucleic acid, RNA, and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80S while the prokaryotic are 70S. Each ribosome has two subunits larger than the smaller subunit. The two subunits of 80S are 60S and 40S while that of 70S are 50 and 30. Here S represents your Febdo's unit, stands for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70S and ITS ribosomes are composed of two subunits. 8.5.7 Cytoskeleton, an elaborate network of filamentous proteinaceous structures consisting of microtubules, microfilaments, and intermediate filaments present in the cytoplasm is collectively referred to as cytoskeleton. The cytoskeleton in a cell are involved in many functions, such as mechanical support, motility, maintenance of the shape of the cell. 8.5.8 Cilia and flagella. Cilia and flagella are hair-like outgrowths of cell membrane. Cilia are small structure which works like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of eukaryotic flagella. The electron microscopic study of cilium or the flagellum shows that they are covered with a plasma membrane. The core, called the exonyme, possesses a number of microtubules running parallel to the long axis. The exonyme usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such arrangement of exonymal microtubules is referred to as 9 plus 2 array. The central tubules are connected by the bridges and is enclosed by a central sheath, which is connected to one of the tubules of each peripheral doublets by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and the flagellum emerge from centriole structure called basal bodies. 8.5.9 Centrosome and centrioles. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles in a centrosome lie perpendicular to each other, in which each has the organization like cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibrin is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is called proteinaceous and called the hub, which is connected with the tubules of peripheral triplets by radial spokes made up of protein. The centrioles form the basal body of the cilia or flagella and spiral and spindle fibers that give rise to spindle apparatus during cell division in animal cells. 8.5.1.0 Nucleus. Nucleus as a cell organelles were first discovered and described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dyes was given name chromatin by Fleming. The interface nucleus of the cell, which is not dividing, has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix, and one of the more spherical bodies called nuclei, singular nucleolus. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bear ribosomes on it. At a number of places, the nuclear envelope is interrupted by minute pores which are formed by the fusion of its two membranes. These nuclear pores are passages through the movement of RNA and protein molecules. Take place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variations in the number of nuclei are also frequently observed. Can you recollect names of organisms that have more than one nucleus per cells? Some mature cells even lack nucleus. Example, erythrocytes of the many mammals and seed tubes of the vascular plants. Would you consider these cells as living? The nuclear matrix of the nucleoplasm contains nucleolus and chromatin. The nuclei are spherical structures present in the nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane-bound structure. It is the site for active ribosomal RNA synthesis. Larger and more numerous nuclei are present in the cell actively carrying out protein synthesis. You may recall that the interface nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin. But during the different stages of cell division, cells showed structure chromosomes in place of nucleus. Chromatin contains DNA and some basic proteins called histones, some non-histone proteins and also RNA. A single human cell has approximately 2 meters long thread of DNA distributed among It's 46, 23 pairs chromosomes. You will study the details of DNA packaging in the form of chromosomes in in class 12th. Every chromosome visible on its dividing cell essentially has a primary construction or the centromere, on the sides of which disc-shaped structure called kinetochores are present. Centromere holds two chromatids of the chromosome. Based on the position of the centromere, the chromosomes can be classified into four types. The metacentric chromosomes, the middle chromosomes forming two equal arms of chromosomes. The submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and the one longer arm. In case of acrocentric chromosome, the centromere is situated close to its end, forming one short and one very long arm, whereas the telocentric chromosome has the terminal centromere. Sometimes a few chromosomes have non-staining secondary construction at a constant location. This gives the appearance of a small fragment called satellite. 8.5.11 Microbodies. Many membrane-bound minute vesicles called microbodies that contains various enzymes are present in both animal as well as plant cells.